Good morning, students. So today we are going to study our next topic, and that is a Chemi-osmotic hypothesis. So student, this hypothesis gives us an idea that how ATPs they are generated during light reactions. As you have studied different types of light reactions, that is cyclic photophosphorylation and non-cyclic photophosphorylation. And what is the common thing between cyclic and non-cyclic photophosphorylation? And that is the generation of the energy-rich molecules, which are ATPs. So how these ATPs, they are generally produced during these light reactions that are explained by the theory, which is popularly known as a Chemi-osmotic hypothesis. So student, if we want to study this theory, let us recapitulate non-cyclic photophosphorylation. As you are well aware that in non-cyclic photophosphorylation, we have two types of photosystem. Photosystem 2, which is known as P680, and photosystem 1, which is known as P700. And there is one reaction, very important reaction that is associated with photosynthesis, and that is the splitting of the water molecule. Okay, student, whenever a particular range of wavelength it approaches this water molecules, as you know very well that in water molecules hydrogen bonding is there. The hydrogen bonds present between the water molecules, it get break. So if we have two molecules of water, then after receiving H new light, that is photon of light, its hydrogen bonds get break and we obtain two protons, that is two H plus ions, along with two electrons and one molecule of oxygen. So as you know, this oxygen gas, it finally released from the mesophyll cells and it later joined the atmosphere. And these electrons, they finally fall into photosystem 2, that is P680. They remain in P680 for some time. And when a particular range of wavelength that is corresponding to 680 nanometer, it reaches the photosystem 2, which is P680, then these electrons, they receive that extra energy. They finally go from their ground states and reach the excited states. But you know that as the excited states, they are the unstable state for these electrons. So they cannot stay there for long. Now what happened? These electrons, in order to obtain again their ground state, they have to release this extra amount of energy. And they adopt a pathway in which they comes in contact with various types of primary acceptors. As you have learned non-cyclic photophosphorylation, so these primary acceptors may be plastoquinone, then plastocyanin is there, and cytochrome complexes are there. So these electrons, they reaches plastoquinone first, and they stay with plastoquinone for some time, and later they supply certain amount of energy to them, and then they reaches cytochrome complexes, which are protein complexes, and also they stay there for some time and release certain amount of energy, and finally they comes in contact with plastocyanin. After releasing certain amount of energy to plastocyanin also, finally most of the energy it lost from the electrons, and these electrons, as they are very less in energy now, they cannot move further and they finally fall into photosystem 1, that is P700. They remain there for some time unless and until they receive the light that is corresponding to 700 wavelength. After receiving the light that is corresponding to 700 nanometer, these electrons, they again get extra energy and they pass through and they do uphill movement, and again they pass through various types of primary acceptors, which are again the plastoquinone, then cytochrome complexes, plastocyanin, and later on the final primary acceptor here is NADP reductase enzyme, and that is associated with the molecule NADP, which is the actually primary acceptor. So when these electrons they comes in contact with NADP, which is the primary acceptor of the pathway related with P700, they gets accumulated there and they get consumed. So addition of electron is also known as reduction. So one thing I want to clear with you that the electrons they gets accumulated, but during the whole course of energy, these electrons they do not use electron carrier for their transport. Instead of that, they use proton carriers. And from how they obtain these proton carriers, so these are actually those protons which are released as a result of splitting of water molecules. So what chemi-osmotic hypothesis tells us that a reaction which is associated with photosystem 2 P680, and that is splitting of the water molecule, when it happens, then some protons, some electrons and oxygen molecule is released. So this reaction, it again takes place on the next day and so on. So as a result, so many protons they are produced, and all of these protons they accumulate themselves, they arrange themselves on the inner side of the thalloid membrane, because non-cyclic photophosphorylation takes place in the thalloid membrane, inside the thalloid membrane. So here one diagram is visible to you. So here what happened, this H2O molecule it gets break and half molecules of oxygen are released and one proton is there and one electron is there. Now, when this reaction takes place again and again on the next day and it repeated in a particular manner, then so many protons they get released, and all of these protons they arrange themselves on the inner surface of the thalloid membrane. So as a result, what happened? As all of these thalloids they are arranged in a fluid-like matrix, which is known as stroma of the chloroplast. So there is a proton gradient established here. As so many protons they get accumulated on the inner surface of the thalloid membrane, whereas the protons are very less outside the thalloid membrane within the stroma. So as a result, a proton gradient it get established. And another thing that I want to explain you that here inside the thalloid membrane, photosystem 2 and photosystem 1 is present, and so many electron acceptors are present. So whenever these electrons they are transported, they comes in contact with various primary acceptors. Then some of the primary acceptors they are present inside the thalloid membrane. Most of the primary acceptors are there inside the thalloid membrane, but there are some primary acceptors such as NADP that are present outside the thalloid membrane within the you can say outside the thalloid membrane on the outer surface of the thalloid membrane. So as the electron they also have to reach this NADP molecule. So again they hire one electron from here and with the help hire one proton from here, and with the help of this proton, these electrons they finally reaches the NADP molecules. They get accumulated at the NADP molecule and the proton is released in the stroma region. So in this way, some of the primary acceptors which are present outside the thalloid membrane, because of them, when the electrons they move outside to reach these primary acceptors, then as a result, some of the protons they go outside from the thalloid membrane with the help of these electrons. But still, the concentration of the protons it is more inside the thalloid membrane, but it is very less outside the thalloid membrane within the stroma. So this proton gradient it get established. Now what happened? Because of this proton gradient, as there are so many protons present here and very less protons that are found in the outer surface of the thalloid membrane, then these protons now they want to go outside by the simple method of diffusion, but the thalloid membrane cannot allow these protons to go. Then finally these protons they come in contact with an enzyme, and that enzyme is the ATP synthase. This ATP synthase enzyme, it consists of two components. One is F0 particle and another is F1 particle. So in our NCERT book, it is CF0 and this is CF1. So these proton molecules, they enter into this F1, F0 particle and they release certain amount of energy here, and as a result of this release of energy, the ATP synthase enzyme, it gets activated, and finally the F1 particle, it releases that particular proton outside into the stroma, and the energy which is released or given by that proton to the F0 F1 particle, that is utilized in producing ATPs. And the same thing is also happen for other protons. So in this way, many protons, they passes through these F0 F1 particles and finally they reaches in the stroma, and later on the concentration of the protons and the concentration of protons in the stroma as well as inside the thylakoid membrane, they later becomes equal at a particular stage. So what happened, this proton gradient, it finally gets break, and in this way the energy is released and that energy activates the enzyme ATP synthase, and as a result so many ATPs that are produced. So in this way, by the process of cyclic and non-cyclic photophosphorylation, these ATPs, they are generated. And these ATPs and NADPH molecules, which are the energy-rich molecules, they are later used in the biosynthetic phase or dark reactions of the photosynthesis. And later, these dark reactions, they obtain energy from these energy-rich molecules, ATPs and NADPH. And as a result, what happened? These biosynthetic pathway or reaction takes place, that is the fixation of carbon dioxide. So 6 molecules of carbon dioxide, it reacts with 12 molecules of water, and as a result 1 molecule of glucose is formed, that is C6H12O6, along with 6 molecules of water and 6 molecules of oxygen are released as a byproduct. So in this way the simple sugar molecules, they are synthesized. So this theory, which is known as the CHEMIOSMOTIC theory, this theory actually gives us a detailed idea that how the ATP molecules, they are generated during these light reactions, whether it may be non-cyclic or cyclic phosphorylation. But one thing you remember in your mind, that these non-cyclic photophosphorylation, here one reaction is associated and that reaction is the splitting of the water molecule. So here one diagram is visible on your screen which shows the chemiosmosis in case of a chloroplast membrane, which is actually the thylakoid membrane. So here all of the components are very much clear to you. So this is a stroma region and these are the thylakoid space. Inside the thylakoid space, the breakdown of the water molecule takes place, and as a result half molecule of oxygen and 1 proton and 1 electrons are produced. So in this way, when this reaction takes place again and again on next day and so on, so what happens, so many protons, they get produced, and all of these protons, they accumulate themselves on the inner surface of the thylakoid membrane, and later they want to try to cross the thylakoid membrane, but that cannot happen, and a proton gradient, it get established on the inner surface of the thylakoid membrane. Now in order to maintain the concentration of the protons somewhat similar on the stroma region also, these protons, they finally enter into the F0 F1 particles, and these F1 F0 particles, they actually contain, they are the components of the enzyme ATP synthase, and they allow these protons to move, but they pass only 1 proton at a time. So as a result, 1 proton, it enters into the F0 particle and give a certain amount of energy to that F0 particle, and as a result what happened, the ATP synthase enzyme, it gets activated and it removes that proton from its F1 component, and the energy which it captures, it later helps in the synthesis of ATP molecules. So in this way, these ATPs and NADPH molecules, they are actually the product of the light reactions, and these later helps in the fixation of the carbon dioxide, and simplest sugars, they are formed from these carbon dioxide. Okay, student, here I have written the entire description what I have taught you. So please go through these diagrams. All of the things are very much clear to you. See how the protons, they form a proton gradient inside the thallophide membrane, and see how the ATP molecules, they are generated by these enzymes which here act as a catalyst. So in this way, two particles are present, F0 and F1. So all the description is there in the CHEMIOSMOTIC hypothesis. So when you view these diagrams and read the whole description, the concept, it becomes very much clear to you. So that's all in today's lecture, student, and this also complete our chapter photosynthesis. I am quite hopeful that the things are very much clear to you. So revise your chapter and read the chapter carefully. And tomorrow we will start our next chapter, and that is cellular respiration. So have a nice day, students.